All right. Okay, so I posted an old exam on eCampus. This is from last year, and I'll go through it a little bit today. Caveat is that this year's exam, as I've said, will be on your computer, so you'll have R available to you. So the types of questions that I can ask will be a little different from what I did last year. But here's what I asked last year. So state the central dogma of molecular biology in your own words. That's fair game. I would like you to know what that is. I'm not asking you to know much more, in the biological setting, but this is very basic, very fundamental. We have DNA, contains information. DNA can be transcribed into RNA. It's like a copy of DNA, and RNA is free to go off and be turned into protein in the process of translation. So we have transcription, DNA to RNA. We have translation, RNA to protein. And the proteins are, for the most part, the molecules that put the DNA into action. But we did talk about other flavors of RNA that do things as well. So in terms of bioinformatics and doing data analysis or trying to answer questions, scientific questions, there's different technologies that we can use to get data of different types. We could look at the DNA itself. What's the sequence of the DNA? What are the structural components of the DNA? There's different technologies that we can use to do that. And that would tell us certain things. If I know the sequence, then I know about the reference manual for an individual. But just knowing the reference manual doesn't necessarily tell us everything about how that information is put into practice. So looking at the RNA copies would give us some different information. Specifically, it would tell me about the activity level of the different genes. Just because you have a gene that codes for a protein, that doesn't tell me whether that gene is on right now. And if it is on, how on is it? Is it pumping out lots of proteins or not? So looking at the RNA copies would be another way to characterize an organism. And then we could also look at the proteins. How active or how many copies are there of given proteins? Each of those ways of looking at the system would give us slightly different information. So just be aware that we have different technologies that do different things. And they're all trying to shine a light kind of at different aspects of this information transfer process. Number 2 was a simple alignment example, pairwise sequence alignment example. And I asked for the table of scores to be filled in. And then based on the table, tell me what the optimal alignment is, or what the optimal alignments are, and what are their scores. So in this problem, I wrote out the algorithm. I figured maybe it would help to remind you, like, what's the actual rule involved in filling in this table. I gave you a set of penalties for mismatches. I gave you the gap penalty, and then this would tell you, this just reminds you, this is kind of a way to write down the rules that we used when we filled in this table. You start with 0, you go to each cell, and you're going to fill in a number in this cell based on the best move you can make. The allowable moves are from the left, upper left diagonal, or top. And if you move from the left or from the top, you incur a gap penalty. If you move diagonally, there's no gap penalty. The only scoring has to do with whether this A matches that G or not. So you can populate all these cells. Each time you populate a cell, you draw a little arrow that indicates did I move from the left or the diagonal or top. You might have more than one arrow coming out of a given cell because there could be equivalent moves. It's just as good to move from the left as it is from the diagonal, possibly. So you fill in all the cells, you add all your arrows, and then once you've got that completed, like this, you can tell me what the optimal alignment is. So in this case, there were two alignments that were optimal. Optimal means I end up in the bottom right cell with this as the alignment score. There were two trajectories that got me here, blue and red. The blue alignment says put this A with that G, because I moved diagonally, that actually lines things up. Then line the next things up, so the C goes with the C. Then put a gap in the shorter sequence, then line up the T's. So the blue says AC gap T. AC gap T aligned with GCAT. The red one, we started by doing 1, 2 gaps on the shorter sequence. Because of the two vertical, two rows that we have gone down before we line up anything, that's 2 gaps on the shorter sequence. Then we put A with A. Then we create a gap in the longer sequence. So vertical moves create gaps in the sequence up top. Horizontal moves create gaps in the sequence on the side. This horizontal move then creates a gap after this A. And then finally, putting the T's together. So the red one corresponds to gap gap ACT aligned with GCA gap T. Both of them had an alignment score of 2. So I would like you to be able to go through that exercise. Fill in a little table like this with a simple alignment problem, 2 short sequences, and tell me what the optimal alignments are. Okay. Number 3, we are talking about an alignment of 2 amino acid sequences. One of them is length P characters, one of them is length Q. We're told that the global alignment algorithm resulted in a score of 19. Okay. Let these numbers, if there are 20 amino acids that are available to make proteins in the human, let's let pi 1, pi 2, pi 20. This problem is about that task of putting a p-value on an alignment, a pairwise alignment. We said we can do this algorithm to do an alignment, but once we've done an alignment like we did in this example, we said there are 2 optimal alignments. There were 2 of them. Both of them had scores of 2. But that didn't tell us anything about was it a good alignment or not. The algorithm will always give you something back, but it didn't tell us anything about how confident are you that it was a good alignment. Because if you gave the algorithm 2 completely unrelated sequences, it would still give you an optimal alignment. So we were interested in putting a p-value on an alignment to tell us how confident we are that it was a real, a believable alignment. So this question is trying to mimic that. I won't ask a question like this on the exam because I will tend to say just do it in R. So this question, if it were on the exam, would be compute a p-value for this alignment and tell me what it means. So suppose we did a p-value. In this case, we had a score of 19. So we did 2 amino acid sequences, and we aligned them to each other. We got a score of 19. If I wanted a p-value, what would... So a p-value is something you compute for saying how believable is a null hypothesis. That's what we do with hypothesis testing. Here's a null hypothesis. I'm going to assume that that's the case. And I'm going to compute this probability, or estimate a probability, that if that is the case, I would see data like mine, or even more unusual data, just by chance. So if I want a p-value for alignment quality, if the p-value is small, like say we do this alignment, we got a score of 19, and the p-value is 0.001, what is my conclusion? Was it a good alignment or a bad alignment? It's a good alignment. That's what we did with these p-values. We said small p-value is indicative of an alignment that you wouldn't expect to happen just by chance. So the null hypothesis when we do this is that the alignment that we just did was just the two things that we were aligning don't belong together. It was just a completely random pairing of sequences of amino acids. So if we see a small p-value that says if that were the case, if these two sequences were just randomly generated without any respect to each other, certainly not coming from the same source, if that were the case, it'd be very unusual to get an alignment score of 19 for two sequences of this length, just by chance. So that would mean I believe this alignment. I don't think this happened just by chance. The way that we did this when we were playing with this exercise was to take one of our sequences, one of these two sequences, and randomly create random versions of it. So if we have a sequence of 8 amino acids compared to 10 amino acids, we have two sequences of amino acids that we're going to align. We get an alignment score of 19. Let's take the shorter sequence and randomly create new sequences that are the same length and that have roughly the same makeup. There's 20 amino acids that we could be choosing from, and in the one that we started with, suppose it's like there's 5 S's and there's 3 P's and so on. Create a sequence of letters where you pick from each of the possible letters with those proportions. So roughly speaking, create a bunch of versions of this short sequence where you've basically randomly picked the letters that go in there. So that if there was a relationship between the two sequences, like one belongs next to the other, they are related, they can be aligned, take that one and basically break any of that relationship by randomly generating letters to define the second sequence. We're creating examples where the null hypothesis is true. Null hypothesis is that the two sequences are randomly oriented with respect to each other. They don't belong together. So whenever I randomly create a sequence that looks qualitatively like this one, in terms of length and composition, when I do that, every time we align to the original longer one, we're getting a realization of this number, a statistic, under the null hypothesis. A p-value is the probability of seeing a statistic value like the one we saw, or even more extreme, just by chance. So we're trying to get that by creating false versions of this second sequence where we know any alignment is a false alignment because it's completely random pairing now between these two sequences. So do that many times, 1,000 times is what we might do, capital B times. Each time you take that fake shorter sequence and you align it to the longer sequence. So each time we're going to get a new score. I saw 19 originally. Now we're going to see 1,000 new ones. And these are all realizations of the score when the null is true. So now we can take those 1,000 scores, like here's histogram of scores, and maybe we see something like this. Scores tend to be located around there. This is what we might call the null sampling distribution of these alignment scores. So then I would put my observed score of 19 up against this and ask how likely would it be to see something that big or even bigger, just by chance. That's the p-value. So I guess I would like you to kind of know that story. You don't, just the idea. What are we doing with hypothesis testing? In the context of sequence alignment, what are we doing? We're getting a p-value. You expect the p-value to be small for a high-quality alignment. Okay? I'd like you to know that. You're going to have your everything with you. You're going to have your R scripts if you want them from the class. You're going to have all the data sets and everything that we've seen in class. So I could ask you, compute the p-value for this alignment. Here's two sequences, compute the p-value. So I would like you to be able to go find the right code, apply it, pull in the p-value, and then tell me what that p-value means. P-value is 0.25, so this is not a very convincing alignment, for example. Go ahead. Homework 2. This one? Okay. Let's see. What was the p-value? 0.118. Okay. So 0.118, how would you interpret that? What I said in the solutions was reasonably strong alignment. And what I mean is that's a pretty small p-value, 0.1. What is a p-value? It's the probability that if it's a completely random based alignment, the probability you'd see this score or even greater. So only 10% of the time. If this was not a real alignment, you could expect roughly 10% of the time to see a 19 or higher. That's pretty small, It's not less than the magic 0.05 threshold. But as we know, there is nothing magic about 0.05. So if you were, you paid a bunch of money to do an experiment and you're going to publish your results, your p-value is 0.06. All is not lost. You have evidence. It's not as small as it could be. So I guess whenever I'm recommending how to interpret a p-value, I would definitely want you to be aware that 0.05 is not magic. 0.06 compared to 0.049, that's not very different in terms of how much evidence there is. So that's what I meant with p-value is 0.11. Okay, it's not less than 0.05, so it's not statistically significant at a type 1 error rate of 0.05, but it's reasonably strong. That's what I mean. Okay. I will not ask you a question like number 4. We really didn't emphasize this very much, but we did talk about the gene ontology stuff. Gene ontology is a way to kind of put at a high level characterize what genes are involved in. And then, so if you have a list of gene ontology terms that are applicable to a set of genes, you can try to ask, is there a term or are there terms involved in that collection of genes that are more prevalent than you'd expect by chance? Can you find gene ontology terms that are enriched in a set of genes? That was another test, hypothesis test, and in this case it was a hypergeometric thing. So Go enrichment stuff that we did in the first script, I think, was dealing with this stuff. I'm not going to ask you about hypergeometric calculations. Definitely ask you something about clustering. Definitely ask you something about principal components, since we've spent a fair amount of time talking about those. Number 5, let's see. I might ask you something like number 5, but I won't make it as long as number 5. This was time-consuming, I think. So what we're doing here is clustering. We're about to do clustering. In clustering, what you need to do cluster analysis is distances. So once you have a matrix of distances between all the things that you want to cluster, you can just pass that into an algorithm. Hierarchical clustering is one algorithm that we focused on mostly, I think, because I think it's the most widely used. So here's a matrix of distances. We have 5 variables. So we have 5 by 5 pairwise distances. Distance between the first variable and itself is 0. Distance between variable 1 and variable 2 is 38.54, and so on. Symmetric. These are symmetric. We're going to do hierarchical clustering, and there will be 4 merge events. We have 5 variables. We're going to put 2 of them together. Now we have 4. We're going to put another, you're going to join again. Now we have 3, again and again and again. There's going to be 4 merge events. The first merge event is going to be go through the distance matrix and find the pair of variables that are most similar, that are closest to each other. So that the smallest distance in here is 37.77, and that corresponds to variable 3, third row, and variable 4, fourth column. First merge will be variables 3 and 4, smallest distance of 37.77. And so this question was looking to make sure you understand what this linkage idea is. Remember, with clustering, there's a couple of important decisions we have to make. One is, how are we going to define distance? And we've seen a few different ways. The default would be the Euclidean distance. The other major piece that we need to make a decision about is linkage. That speaks to how do we define distance between clusters. If there's 2 clusters and each cluster has multiple individuals in it, how do we define distance between them? So, remember there's 3 different types. There's complete, single, and average linkage. Using complete linkage, tell me about what the clustering algorithm is going to do. The first merge event is 3 and 4 coming together because of their distance of 37.77. What are the second, third, and fourth merge events? When we put the third and fourth variables together, there's now, instead of 5 entities, there's 4, because 2 of them have been joined together. So after we have done that first merge, we need to recompute distances so that we can take the next step. I need a 4 by 4 matrix of distances now. And the way I've ordered things to go into this 4 by 4 matrix is step 1 was take variables 3 and 4, put them together. I have put that new cluster containing 3 and 4 in as the first variable now. So the first row and the first column, the first row, first column corresponds to this new cluster containing 3 and 4. This 42.72 then is the distance between this cluster containing 3 and 4 and the, quote, cluster containing just variable 1. And this 44.48 is the distance between 3, 4, and 2. 49.65 is 3, 4 compared to 5. But now I'm again just looking for who's closest at this point. And the smallest number is this 38.54. That's the same 38.54 from here. That's the distance between variable 1 and variable 2. Down here, it's the same thing. It's row 2, column 3. Thing 2 is variable 1, and thing 3 is variable 2. So I'm going to take variables 1 and 2, and they go together now. So let's see. Smallest distance is row 2, column 3. That corresponds to a merge of variables 1 and 2. So that was the second step. We've done 2 merges now, and we need to move to the next step. Now I have 3 entities. I have a cluster containing 3, 4, a cluster containing 1 and 2, and then 5 is left off by his or herself right now. Now I need a 3 by 3 matrix of distances, and it's the same game, same approach. The distance between 3 and 4 and 1 and 2, for example. So we have 2 entities, it's 3 and 4, and it's 1 and 2. Let's match this number, by the way, just so we're sure we know how to match these things. 44.48, where did that come from? It is the distance between 3, 4. So it's a distance between 3, 4 and 1, 2. And since we're doing complete linkage, it's going to be the maximum of 3 with 1, 3 with 2, 4 with 1, and 4 with 2. So basically, we have 2 clusters. There's 3 and 4, and there's 1 and 2. Linkage says compute all the pairwise distances across those clusters. So 3 to 1 and 3 to 2, 4 to 1 and 4 to 2. Compute all of those distances. And if you're going to do complete linkage, you take the biggest of them. Let's see if we can match that. Over here are the original distances, and I want to do 3 and 4 compared to 1 and 2. 3 and 1 is third row, first column. That's 42.37. So this is going to be max of 42.37. 3 and 2 is 43.86. 4 and 1 is 42.72, and 4 and 2 is 44.48. So maximum distance is this 44.48. That was the biggest. That's right. Yeah, that would be faster. So it is 44.48. I'm sorry. It is 44.48. Could have, which one were we doing? Are we doing this one? Yes. 3 by 3. Okay, so this is the way we calculated this 44.48 was kind of first principles and referring back to the original distance matrix. Since we have already made this 3 by 3 revised distance matrix, I could have just looked at this. So these are the distances. Where's the biggest, or where's the smallest number now? It is 44.48, which is the distance between 3, 4 and 1, 2. But you know how to go back and see this in terms of the original matrix also. Okay, good question. So suppose we come up here, let's start out, and we have all these distances, and there's a tie. Suppose 38.54 and 38.54. I think that will depend on the software package that you use, but what R does is it picks one. Actually, there's some interesting details here. It is possible that you would put them all together at once. So if these two are close and those two are just as close, depending on the nature of the data, and it's a little bit complicated, it's possible that they will all go together in one move. Most of the time, it will, like R will just pick one of these two, and then it'll just repeat. Chances are the next move is going to be these two. But most of the time, you only join two things at once. So here we are down at, we're almost done. We said here's our new distance matrix. And now who's closest? It is the smallest number in the first row, second column, 44.48. So next up is to join those two clusters. That's the cluster with 3 and 4 and the cluster with 1 and 2. So next move is to create this. I have 1, 2, 3, 4, and 5. This is the distance now. I would again apply that complete linkage. What's the distance between 5 and 1? 5 and 2, 5 and 3, 5 and 4. Take the maximum. 5 and 1, 2, 3, 4. What's the maximum? 49.65. Okay. Given all of that you just figured out, you can draw things. So here's the dendrogram and scree plot. Dendrogram. Is that it? No. That's not it. Dendrogram, scree plot. 3 and 4 were joined first, and the height, the merge distance between those was right around 38. Then it was 1 and 2, and then the next move was to put 1 and 2 together with 3 and 4. So there's this joint, and that merge height was the 44 point whatever, 44.48. And the final merge was to put them all together with a height of 49.65. Scree plot is just a plot of the merge heights. So the first merge, 1, had a height of 38-ish. The second one was a little bit higher because those were a little further apart. Third, fourth, that was the final one. And I'm looking at this picture and trying to say, is there any obvious elbow, where there's a big jump? As we made this joint, it was hard to make that joint because these things were very different from each other. Can I find places like that in the scree plot? And I might say this is where I'm going to draw the line because of this vertical distance being, relatively high compared to that vertical distance. So I might kind of, in my mind, draw a horizontal line right there. That's where, below which, I think the joinings constitute real structure. So if I did that, there would be a cluster and a cluster, because those have not been joined at this point. And this 5, I would envision it dropping all the way down because it has not been joined with anything either. So if I pick that, that's the place where I think we caught the real structure. That's corresponding to 3 clusters. 1, 2, 3. Number 6. We have 10 samples. 5 of the human samples had colon cancer, 5 of them were healthy. And here is the quote metadata for this experiment. Samples 1 through 3, 4, 5. So the first 5 columns of the Excel spreadsheet, those are cancer people. Then the last 5 are normal people. We're told that the samples were run in batches, which is not uncommon. It takes 4 hours to run a sample through an instrument maybe, so you physically can't run all 10 on the same day. So we will run 3, we'll run a few today, and then we'll run a few next week or something like that. We have a batch variable that tells me the batch number that each of these individuals will run in. So the first 2 cancer patients were run in batch 1 together with 3 individuals from the normal group. Second batch had 2 cancer, 1 normal. Third batch had 1 cancer, 1 normal. We also are told that there were 2 technicians who ran these or prepared these samples, and we're told who they were: A, B, 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 A, B's. Okay, and then figure 1 shows clustering results for these data. Euclidean distance and complete linkage. Tell me about the picture. What do we see? Here's figure 1. How many clusters would you say there are? I would say 3, because that seems to be where the first real jump happened. So that would correspond to 2, 3 clusters. I would draw an imaginary horizontal line right there, which would mean this is its own thing, that's its own thing, and that's its own thing. So based on this picture, I think there is some structure to the data. And what would I say about it? Here's 1, 2, 3, 4. What do I know about them? Those are all cancer people. 6, 7, 8, 9, those are all normal people. So this appears to be structure that's maybe a difference between the comparison groups, cancer versus normal. That is completely sensible, makes sense, that is not worrisome, that kind of makes us think the data are doing what we would expect. This is a little worrisome. Individual 5 clustered together with individual 10, and quite a bit different from everybody else. So this pair is unusual when compared to everybody else. What do we know about 5 and 10? Batch 3. Okay, so 5 and 10. If I were looking at that clustering result, I'd come back to what I know about the way the experiment was run, and the common denominator between 5 and 10 is that they were both run in batch 3. They were the only ones that were run in batch 3. So that casts a question mark on batch 3. What was unusual about batch 3? Anything? Is it, yeah, could have been anything. We recalibrated the instrument that day, or we had to switch the reagents. We ran out of one bottle of reagents halfway through, and we had to refill it with another batch, so there might have been some issues there. Air conditioner went out during the afternoon while we were running this stuff. Could be anything. Part B, we have cancer and normal, and let's go test peptide by peptide. Is this peptide different comparing cancer to normal? So 2-sample t-test was applied to each of the 10,000 peptides. Figure 2 contains a histogram of the resulting p-values. So based on the histogram, does that look like what you would expect it to look like? Does that look like a reasonable, valid histogram of p-values? That's the pattern that we said is kind of what we expect. Remember that we've said we think of these histograms of p-values, like the one we just saw, as a mixture. There's some proportion of uninteresting features floating around, and what's left is interesting features. So remember, uninteresting features are features for which the null hypothesis is true. In this case, the null hypothesis is that the mean intensity for a peptide in cancer is the same as that in control or healthy. The distribution of p-values for those types of peptides is uniform between 0 and 1. For the truly differential peptides, we expect to see smaller p-values systematically because there is a difference. So I expect to get small p-values. So based on the picture, that I don't see anything worrisome. There's not like a weird pattern to it. I see that there is, a fairly substantial peak over at the left, which suggests that there's a fairly substantial contribution here. So there are a fairly substantial number of differentially expressed features. And in the exam, I asked you to comment, would you say that there are differentially expressed peptides, yes or no, and why or why not? What I was fishing for there was, yeah, it looks like there are a fair amount because the histogram is definitely not flat. If it were completely flat, I'd confidently say there's nothing in these data. If it's barely not flat, like that, maybe there's one or two interesting features, but that's not very convincing. Not a lot of signal or structure signal. Number 7 is SVD stuff, and I have de-emphasized SVD for us. That said, I could easily turn this into a principal component problem. So let's see if we can do that. We have new data. These are time course data. We have 5 samples of E. coli, and each of the samples, for each of them, we created a sample, like an array or whatever technology we're using, at 6 equally spaced time points. So it's time course data. It's correlated data, longitudinal data. At the first, third, fourth, fifth, and sixth time points, the samples were stored at 30 degrees centigrade. But at the second time point, the temperature was raised briefly to 42 degrees centigrade. The idea here is to take E. coli and shock them, subject them to heat shock, and see what happens. So we'll measure them right now, and then in an hour or something, we'll spike the temperature. And then we'll measure them every hour, or probably not an hour, every few minutes after that. Okay. Yeah, let's not use this as an example right now. This is more SVD specific. So for the purposes of the exam, don't worry about SVD, but do worry about principal components. So know how to apply and interpret principal components. Should you standardize the variables, yes or no? Think about that. We've talked about that. Should you standardize the variables, yes or no? My stock answer is, why not just always standardize them? If you don't standardize them, you can get kind of weird results. In particular, if you have variables that differ greatly in scale. Like I have a variable that's the proportion of something between 0 and 1. And then the next variable is income in dollars for the year. Huge difference in scale. If I just did PCA on the untransformed, unstandardized variables, all PCA is going to say is that income drives everything, because it's going to be looking at just unscaled variability. Where's the source of the biggest variability? And if there's big differences in scale, it's just going to be the variables that have big scale. When you standardize, you turn the variables into all having mean 0, all having standard deviation 1. So now that differential scaling stuff is not relevant anymore. And what PCA is going to tell us is about co-relationships. Tell me about structure in the data that's driven by co-relationships between the variables. You can tell me what proportion of the variance is explained. You can tell me something about the pattern that drives that explanation by looking at the eigenvectors. Okay. So I would say review what we've done in class on PCA. Make sure you're comfy with how to apply it, how to interpret it. Not going to go deep into the mathematical details of it, so I'm not going to ask you to compute anything manually, certainly not. I might ask a little technical questions that I don't expect everybody to get, just to separate A plus from A, that kind of thing. But don't feel like you have to be prepared for lots of technical things. All right. Let's see. I was going to the data. There's going to be at least one data set. There might be 2 that are required on the exam, and I'm going to post those over the weekend on eCampus, and I will announce when they're available, just in case you're concerned about not being able to download the data during the exam. Mostly for people at a distance who will be using a proctoring service. If they want, or if anybody wants, you can download the data that we're going to use over the weekend. I got to go find the data first. I'll go find some data, post them on eCampus. You can download them if you want to. And then for the exam, you can bring whatever you want. So computer, notes, phone, you can access the internet. You will need R to do some analysis. So you're going to download a PDF from, well, actually, I'll hand out, in class, I'll hand out a piece of paper. If you're at a distance, you'll download a PDF. And then what I would like you to do, everybody, is open a Word document, type in answers, to text-based questions. Do screenshots of maybe a picture. If I ask for a figure, go to R, make a picture, do a screenshot of it, paste it into Word. Save the Word file as a PDF and upload that to WebAssign. Wondering about WebAssign. Do you guys, you do not use WebAssign at all? Okay, so local students do not use WebAssign. So what I'll have you guys do, I think, is just email me. So by the end of the class period, I need to have had a time-stamped email from everybody in the classroom with your PDF submission. Is that confusing or worrisome to anybody? Email me. So if you're at a distance, then you will upload through WebAssign like you usually do. If you are a local student, you're still going to have a PDF, but just email it to me. All right. Questions? This long? This was probably too long, right? I will try to make it as reasonable as I can. But again, if it's a 90-minute exam, I won't mean to make it a 90-minute exam, but it's not always easy to make exams. If I do mess up and make a 90-minute exam, it's a 90-minute exam for everybody, which means do the best you can, finish as much as you can. I'm not going to fail everybody. So. All right. Yes. Yes, solution for homework 3, I'm doing it right now. I'll post it today.